to all. Myself Param Malik, a student of BSc Honours Chemistry currently studying at the Kirodimal College, and today I am here to even better make you understand the topic amino acids and their classifications. So before starting the topic, we should have a clear roadmap in our mind for what we have to study in order to have a mastery on the topic of amino acids. So we should be able to define amino acids, what are amines, the carboxyl groups, the side chains. We should understand the topics of non-polar, polarity, acidic, basic, the aromatic compounds, and also we should be able to interpret pKa and ionization. So L versus T isomers, or we can say levo versus dextro rotatory isomers. We should have a basic understanding of these topics. So the basic roadmaps includes bonding, stereochemistry, their classifications. The classifications are based on various aspects as we will see further. So without delaying, let us continue. Going to the next slide. What are amino acids? Their fundamental structure and nomenclature of amino acids. As we can see from the name amino acids, amino means amines and acids is basically acid. So amino here stands for the NH group, and acids can be referred to as the carboxyl group. So when we merge together with the help of a side chain, we get amino acids. As we can see the structural diagram, NH2CHR. R here refers to the side chain and COH is the carboxyl group. When we merge them together, we get an amino acid, zwitterion at physiological pH. So first of all, what is zwitterion? It is nothing but a compound having both positive and negative charges. For example, NH3 plus and COO minus. It has both positive and negative charges, so it is considered a zwitterion. So when studying about the basics, we should know from the start. So the first topic, nomenclature, how we should write their names. We should count the number of carbons depending the priority, or starting from the priority of the higher group. From here we will start from the carboxyl group. So this is the alpha carbon, so we can write 2 amino. This is the second, which is linked to the amino group NH2, 2 amino carboxyl COH group acids, because it is of course acid, and acid has higher priorities than amines. So yeah, it is the acid. There are also several classifications or standard terms used to refer to these amino acids. These can be 3 letter or 1 letter. For example, a simple non-polar structure, glycine, also represented by GLY, or we can simply use G. Alanine, we can refer it by A or ALA, or serine, we can refer it by SER or S. Keynotes. Okay, glycine is an exception, except all the all the amino acids are chiral in nature. And just to keep in mind, side chain defines chemical properties and classification. Because the carboxyl group and the amino groups are almost the same, it is only the side chain that binds them together which determine their physical properties and also the chemical ones. So next topic is peptide bond formation, condensation and primary protein structure. What is a monomer? Well, you can see 2 alpha amino acids each with NH2 and COH functional group, like 2 alpha amino acids. When they go through condensation, carboxyl of one reacts with amine another, meaning N terminus reacting to C terminus. What is N terminus? N terminus can also be referred to as the starting terminus. It is basically referred to the amino terminus, like NH2 group, and the C terminus is basically referred to as the ending terminus. It is basically referred to as the carboxyl group or the COH. Well, forming a peptide bond, the COOH of the first compound reacts with the N terminus of the second compound, so they are bonded by peptide bond. or we can also say they are bonded by amino acid linkage. When we continue this process, we will get a big chain, also known as a polymer. So when we talk about a single compound, it is known as monomer. Like glycine, it is a monomer. Alanine, it is a monomer. But when they combine together through N terminus to C terminus bonding with the help of NH2 and COOH group, they can be termed as amino acids, or more clearly, they are binded by amino acid linkage and are called as peptides. So, going on to the next slide, we have stereochemistry of amino acids. Basically, they have L and D isomers. These are nothing but the rotation. Levo stands for L and D stands for dextrorotatory. Levorotatory and dextrorotatory isomers, it is nothing because just a simple concept. If a compound has chiral carbon, it will always rotate on either side. So glycine is the only exception, as we can see, because it has a chiral or it is not optically active. So what does chirality at alpha carbon mean? Most protogenic amino acids are chiral. Glycine is a chiral, as I have told you before. L versus D configuration. Proteins use L amino acids, Fischer projection. Fischer projection, it shows NH3 plus on the left, and for better visual models we can use wedge-3D as you have learned in your previous semesters. Just remember, glycine is the only exception because it is optically inactive. Going on to the next slide, we have amino acids and their classification. So this is the basic list of amino acids that are majorly used. There are many other amino acids, but these are the majorly used, which have 3 letter and 1 letter, that are mostly responsible for all the major reactions. When we go through it, they are non-polar, polar, acidic or basic in nature. We will study it later in the video. So alanine, this same word has a 3 letter abbreviation ALA and also a 1 letter abbreviation A. Arginine, asparagine, aspartic acid, cysteine, glutamic acid, C. Some abbreviations have similar wording like GLU and GLN, glutamic acid and glutamine. You can be confused between them, but there is a basic difference. One is acid while the other is not. They are similar, you can be get confused, so try to understand them better. So there are many like leucine, lysine, proline, serine. They are all different depending on the side chains they are linked with. Valine. So these are the basic 20s that we have to keep in mind. Going to the next part. So first type or first classification of amino acids which we can have are known as non-polar. Non-polar means they don't have positive negative charges at the end, or they are hydrophobic. See, if they are not charged at the ends, so they will not react with water, so they are hydrophobic. The best examples are the residues because they will not react, so they will not be soluble. If they are not soluble, then they will be a residue. Best examples are glycine, alanine, valine, leucine, isoleucine, methane, prolinium. These are the examples. Properties: non-polar side chains like strong hydrogen bond donors. They are not bonded by hydrogen bonding, so they definitely lack strong hydrogen bond. As we said, these are the non-polar side chains. They prefer interior of globular proteins, dry folding via hydrophobic effect. They have no hydrogen bonding to keep them stable, so they sometimes fold into a hydrophobic effect. They also, so some functional notes for the same: aromatic residues like the amino acids having an aromatic ring enable stacking interactions. Okay, they are good for enable stacking interactions because of the ring. TRP is a strong UV absorber and used in spectroscopy. This is a functional note because it is an important category. because most of the amino acids are does not met the condition, and also it has a methyl ether often buried but can be surface exposed. So these are some of the known polar hydrophobic amino acids. Now the second classification falls under the polar uncharged amino acids. When we say about polar, they have charges at the end. If they have charges, they will react with water, and if they react with water, they form hydrogen bonds. So they are pretty much stable and they do not form into like or fold into globular structures. So residues, or basically examples for the polar uncharged amino acids are threonine, serine, glutamine, tyrosine, commonly polar. Okay, properties: side chains form hydrogen bonds, as we have already seen. Increase protein solubility. When they form bonds, they stabilize the structure, hence increasing the whole peptide linkage, making them strong, soluble, and stable. So functional notes: serine, threonine, OH act as a nucleophile. This can be covered under the process of serine proteases, or more prominently serine proteases catalytic triad. Stabilized structure: asparagine, glutamines stabilize via hydrogen bonding. Tyrosine is both polar and aromatic. Cysteine can be polar. These are some of the functional notes which you have to remember. Now going on to the next category, we have acidic and basic amino acids. So classifications: first of all, let us remember there are 20 amino acids. Some fall under the category of polar amino acids, non-polar amino acids, or we can call them hydrophobic and hydrogenic. Classifications, and the third and fourth classification are basically acidic and basic amino acids because they depend on the pKa values and ionization. So the best examples, or the most suited examples for these acidic and basic amino acids are residue: aspartic acid, glutamic acid, lysine, arginine, histidine. They have a certain pKa values of their side chains, as we can clearly observe. Physiological form: deprotonated, negatively charged, so it is present at the C terminus with the COO minus. Also negatively charged, present with COO minus at the carboxylic end. But for lysine, it falls under the basic category. So notice there is a plus charge on the amine side, which is the NH group. Arginine, also the basic, protonated, positively charged, it will have more positive charges as compared to the lysine. And histidine is 6, partially protonated, near physiological pH. It is also near the neutral pH, so there's a little difference to it. Going to the next slide, we have special case amino acids. We have to always remember when we study about chemistry, organic, inorganic, especially organic, there are always exceptions. So what are those exemptions? First one, aromatic, sulfur containing, and proline. Structural roles and key facts: we have the aromatic ones like phenylalanine, tyrosine, tryptophan. They can absorb the UV rays. TRP is strongest at 280 nanometers, so the ring provides them an edge to absorb the UV rays, which currently is lacked by the other amino acids because they can't process it without the ring. Sulfur containing: cysteine, thiol forms disulfide bonds, stabilizing tertiary structure. It has its own forms. It is experimented and proved that cysteine in when currently in thiol form stabilized tertiary structure with disulfide bonds. When we talk about methine ion, its thioether serves as start codon in protein synthesis. It is generally observed, so it's a kind of a property. Proline, secondary amine that imposes conformational constraint, introduces kinks in helices. It is observed there are two kinds of helices, basically a single strand helix or the double strand helix. When we say more than one helix, these are helices. So secondary amine that imposes conformational constraint, introduces kinks in helices. So yeah, They are good for the team. They do good work, so they allow and introduce kinks in helices. So coming on the next topic, it is one of the most essential or non-essential amino acids. Is the one of the most important classifications. So before going into the classification, we need to understand what does essential mean or what does non-essential mean amino acids when it comes to a human body. This is a topic of a bioorganics, so we are talking in regards of the human body. So essential that essential amino acids which can't be produced by a human body. For example, histidine, isoleucine, leucine, lysine, they can't be produced by the human body and thus need to be obtained via food. Adults require alanine diet, methionine and phenylalanine serves as precursors, whereas the non-essential ones are the one which can be produced or synthesized by the human body. For example, alanine, asparagine, aspartate, and so on. Tyrosine synthesized from phenylalanine. Some become conditional during stress. So yeah, some non-essential or which are synthesized by human body are also required in important aspects of a human. Some deal with stress too. So they are important for the human body, so they are non-essential ones. Classification, summary table, amino acids grouping. We have basically classified, let's start from the start. We have 20 amino acids. We have differentiated them based on the known polarity, acidic nature, basic nature, their aromaticity or their aromatic behavior, basically containing a ring. Sulfur containing disulfide bonds can be formed pretty much easily and stable. Essential, like dietary requirement for adults. So these are the most important classifications that we need to study about the amino acid classifications to have a mastery on this topic. There are certain things when we talk about the non-polar are hydrophobic, do not form hydrogen bonds. Polar charged, basically they form strong hydrogen bonding. They also have active sites role. Acidic, negatively charged because of the pKa values. Basic is positively charged also. Their pH is above normal. Aromatic, UV absorbance, stacking interactions. UV absorbance, best DRP is at 280 nanometers, which is quite good. Sulfur containing, they form disulfide bonds. Again, essential dietary requirements for adults. These are some of the mentioned essential amino acids that the body need to consume. So coming to the next slide, we have structure to function. Yeah, there are several things like amino acids do not act in isolation. They are dependent on each other and react with each other to form stable systems. One of the systems is serine protease catalytic triad. SCR, also referred as serine, acts as a nucleophile. Histidine as base and aspartate stabilizes histidine. So they not both, all the three of the amino acids serve as a purpose for the other and they all three stabilize each other. The best example is chymotrypsin. The second structure to function case study we have is insulin disulfide stabilization. Cysteine pairs form inter and intra-chain disulfides critical for hormone activity. It means cysteine pairs intermolecular bonding and intermolecular and intramolecular. It means in the molecule and between the molecules to form disulfide changes. Hydrophobic code mutation. It is the third type of structure to function case studies that we are going to study. So hydrophobic co-mutations. When we talk about the peptide linkage, as we have talked before, some are hydrophobic in nature. It means they do not form hydrogen bonds, so they can be replaced by the polar residues which do form hydrogen bonding, so they better stabilize the structure and impairs the function which was disabled by the before hydrophobic amino acids. So coming on to the next slide, we have study strategies and mnemonics for amino acids. So, here's a quick my experience how to learn them better and memorize them better, not to cram it but to understand them. First, group mnemonics like have some games like writing random number letters PVT, Tim Hall, and then making reference to them like P stands for phenyl, V stands for valine, and T stands for threosine. Similarly, connect them using the C and N terminus for better clarity of the structures, active recall, and spaced repetition. You can make some games based on flashcard for the names code structure. The one matches wins the game, or the one who guesses wins the game. It can be played weekly. We can practice doing structures like sketch. They should be at the back of our head. Now, suggested problems: predict ionization states at given pH values. This topic may take a time, but it will come to you if you practice it regularly. So these are the 4 steps which I would recommend to understand this topic better, and yeah, to have the best of the knowledge on this topic. So key takeaways. What we have learned: we have learned about the amino acids and its classification based on many terms like how they are essential, non-essential, non-polar, required by the human body, produced inside the human body, have to be taken from the external source via food, how they are non-polar in nature, polar in nature, how the hydrophobic ones can be replaced by the hydrogen bonded ones so that the procedure may continue, which was before disabled. We have also studied how they differ depending on the pKa and charge values, that is basic and acidic, they behave differently. Special residues, we have learned about the disulfide bonds and the property to absorb UV. Don't forget TRP is best at 280 nanometers. Further reading, these are the better references for better knowledge and to get a better grasp on this topic. So thank you, that was all for today. Hope you understand the topic. Thanks a lot.